Hello everyone and welcome to the channel. Today we are diving into the fascinating world of viral diagnosis. When a doctor suspects a viral infection, how do we confirm it in the lab? We are going to break down the key methods from classical techniques to the modern molecular powerhouses. Let's get started. I am Dr. M. Manikandan, Assistant Professor from the Department of Microbiology, Sacred Heart College, Tirupattur. Introduction. It is very much essential to diagnose viral infections for the proper treatment procedures. There have been many new developments in the laboratory for viral diagnosis, which provides more rapid and sensitive viral identification from the collected clinical samples. These procedures include better antibody reagents for the direct analysis of samples and also the molecular level techniques along with genetical identification and genome sequencing for direct identification of the virus and assays that can identify multiple serotypes of the virus, which is to be automated. The diagnostic toolkits. We can group the main methods of viral detection into four key categories as different tools in a detective's kit. Some look for the virus itself, some for its parts, and some for the evidence our own body leaves behind. The first method of detection is the virus isolation and growth. Second is the detection of viral proteins and viral genomes. The final method is the serological analysis of antibodies. Viral laboratory studies helps us to confirm the diagnosis by identifying the viral agent in an infection and determination of appropriate antiviral therapy to the infected individual, checking on the compliances of patient taking antiviral drugs, defining the course of the disease, and finally monitoring the disease epidemiologically and educate the physicians and patients. These are the various essentials of a viral laboratory assay. Specimen collection. For every laboratory diagnosis, the sample collection is an important step. The patient's symptom and history, including recent travel, the season of the year, and a presumptive diagnosis, helps determining the appropriate procedures to be used to identify a viral agent. The second step is the collection of specimen from infected. The samples may be collected from respiratory tract, gastrointestinal tract, maculopapular rashes, vesicular rashes, central nervous system, that is cerebrospinal fluid, urinary tract samples, then blood, which is one of the important sample containing antigens and antibodies. These are the various samples we collect from infected individuals. And specimens should be collected early in the acute phase of infection before the virus ceases to be shed. The respiratory tract sample should be collected from 3 to 7 days of infection, especially this herpes simplex virus, that should be collected less than 5 days of infection. The first method is the cytological examination with the golden standard procedure, that is virus isolation and cultivation in tissues or in animal models. We identify this cytological effects. Many viruses produces characteristic cytopathic effects. The characteristic cytopathic effects in tissue sample or in cell culture include changes in their cell morphology, cell lysis or vacuolation, In some cases, syncytia formation and inclusion bodies. Syncytia are multinucleated giant cells formed by viral fusion of individual cells. Tens or hundreds of cells will become fused to form a individual cells where it will contain multi-nucleus. Example includes paramyxovirus infections, herpes simplex virus infections, varicella zoster virus infections, and HIV virus infections. These viral particles promote syncytia formation. The inclusion bodies are either histological changes in the cells caused by viral components or virus-induced changes in cell cultures. For example, intranuclear basophilic inclusion bodies, that is otherwise called as owl's eye formation, found in large cells of tissues with the cytomegalovirus infections. The most pragmatic identification is the rabies virus formation as an agribody in the brain tissues after an rabies viral infection. These images provides us a detail of a syncytia formation, that is a multiple number of cells fused together due to the effect of a virus infection, and it becomes a multinucleated giant cell, otherwise called as a syncytium, which holds numerous number of nucleus and also viral particles. The second image shows us the negri bodies, that is formed after an rabies viral infection, which is found in the brain tissues of the infected individual. Virus isolation and growth techniques. A virus can be grown in tissue culture, embryonated eggs, and experimental animal models. Embryonated eggs are still used for the growth of virus for some vaccine production. They have been replaced by cell cultures for routine virus isolation in clinical laboratories. Experimental animals are rarely used in clinical laboratories for the purpose of isolating a virus. In tissue culturing techniques, primary monkey kidney cells are excellent for the recovery of influenza viruses, paramyxoviruses, and many enteroviruses, along with some adenoviruses. The human fetal diploid cells are generally fibroblast cells, support the growth of a broad spectrum of viruses, including herpes simplex virus, varicella zoster virus, cytomegaloviruses, adenoviruses, and picornoviruses. The HeLa cells, otherwise called as the continuous cell lines of epithelial cells derived from human cancer, which are all excellent for the recovery of respiratory syncytial virus, adenoviruses, and HSV viruses. Viral detection. After the collection of sample from the infected individual and cultivating the viral particles into a cell culture, how can we identify the or detect the viral growth? A virus can be detected and initially identified through observation of the virus-induced cytopathic effects in the cell monolayer by immunofluorescence techniques or genome analysis in the infected cell culture. For example, a single virus infects, spreads, and kills surrounding cells, which are otherwise called as plaque formation. The type of cell culture, the characteristics of cytopathic effect, and the rapidity of the viral growth can be used to initially identify many clinically important viruses. Quantitative detection of virus. The viral load in the infected individual can be quantitatively analyzed by determining the greatest dilution that retains the following titer values. The first value is the tissue culture dose, TCD 50. which shows us the titer of virus that causes cytopathological effects in half the tissue culture cells. The second one is the lethal dose, that is LD50, in which the titer of virus that kills 50% of a set of test animals. This shows us the LD50 values. The third one is the infectious dose, that is ID50, which is titer of virus that initiates the detectable symptom, antibody, or other responses in 50% of a set of test animals. The number of infectious viruses can also be evaluated with the count of plaques formed by tenfold dilutions of the sample, that is plaque forming units. Serological tests. The enzymes and other proteins produced during viral replication can be detected by biochemical, immunological, and molecular biological means. The viral proteins can be separated by electrophoresis, and they are patterns used to identify and distinguish different viruses. Genome amplification tests, including PCR for DNA genomes and RT-PCR for RNA genomes. These are the methods of choice for detection and identification of viruses. The indirect fluorescent antibody test and solid phase immunoassays such as latex agglutination and ELISA are commonly used to detect and quantitatively analyze the viral antigens and antiviral antibodies. Thank you for watching. Subscribe for more simplified science. Comment your questions below in the comment section. Thank you again.